All right, we're recording. It is Friday, April 14, starting on slide 26, and we are going to be going through this faster. We're going to try to get as far as we can possibly finish today. I want to get us on the urinary system as quickly as possible. Quick reminder, quiz is on Wednesday. You have just the handout for homework that's due on Wednesday. If you choose to do the coloring, it is optional and extra credit, just as a reminder. I did put on Moodle what you need to know for your muscle quiz next week in lab, and I emailed it to everyone as well, so check that, okay? There's one dissection video up there now and all the pictures, and I will continue to add more videos, okay? So, in the meantime, study your minks, get your list, you're doing a good job. Okay, I heard that overall grades were pretty good on the last quiz. I haven't personally looked at them. My TAs just kind of mentioned it to me, so I'll look at them, and I'll look at the muscle quiz from this week. I did promise to curve that one. So please remember that right now, whatever it is. It will probably improve when I get to look at those grades later today or later this weekend, okay? All right, so let's get going. We talked about hormones, but we didn't really talk about what they did, so now that's where we're at. So functions of hormones in the GI tract. Remember, there are endocrine cells within the stomach itself and the pancreas and fun stuff. So first of all, gastrin. Gastrin is secreted by the stomach. What stimulates the secretion of gastrin? So distension. So you've been eating your food, filling up your stomach, proteins that are starting to be digested, autonomic stimulation, so by the vagus nerve, okay? Alcohol or caffeine will also stimulate it. So we don't drink alcohol. I do drink coffee in the morning. So some people, if you drink coffee on an empty stomach, because coffee is acidic, can upset your stomach because that stimulates the secretion of even more digestive hormones and enzymes, which can make your stomach a very unhappy environment if you don't have food in it. Okay, so secretory. So if you have gastrin secreted, remember it travels in the bloodstream and comes back and tells the stomach to secrete even more. It doesn't cause any movement, so motility is talking about smooth muscle movement like peristalsis or mixing movements. That doesn't have anything to do with it. The duodenum, the first part of the small intestine, secretes secretin. What a nice name. And secretin is stimulated when the chyme from the stomach enters the duodenum and that pH is less than 2. So that was on one of our little graph pictures we looked at on Wednesday. So if the chyme, that pH, let's see, is less than, did I do that right? pH is less than 2? Wait. Yeah. What? Oh. Less than 2. Yes. No, I need to write that right. pH is less than 2. Did I do the little thing in the right direction? I think so. Okay. Secretory. So if secretin is released, it decreases gastrin. Okay. So it's a kind of a negative feedback reaction with gastrin. But it stimulates secretions from the pancreas and bile, which is coming from the gallbladder. And it decreases gastric motility. So if the stomach is churning, if secretin is secreted, it's going to slow down that movement, okay? Then we have cholecystokinin, which is also from the duodenum. This is one of the others that is... This is important for breaking down fatty acids and peptides. So peptides are proteins, okay? And let's see. It has a little decrease on gastrin, but look at stimulating pancreatic secretions and gallbladder. So secretin and cholecystokinin have very similar functions, except for when we look at the motility, cholecystokinin actually does have a strong effect on the motility, strongly decreasing the movement within the stomach, okay? So similarities and differences. So looking at the small intestine, there are 3 sections. You must know those 3 sections of the small intestine in the order in which they come. And also, this very important fact that's in bold and italicized. The small intestine is where you have the most nutrient absorption and water absorption. This is where the majority of it all happens, is the small intestine. Yes, water absorption does happen in the large intestine, but most of it is happening in the small intestine, okay? So the duodenum, this is that first portion of the small intestine right after the stomach. So right there, you're entering the duodenum. Duodenum's it's not very long. It says 25 centimeters, so yeah, it's not that long. Then the middle section is the jejunum, so kind of like the middle section where it's all folded in, and this is long, 2.5 meters long. So a meter is approximately 3 feet, approximately. So you could say it's like 7.5-ish, 8 feet long or so. It's pretty long, okay? And then the last portion is the ileum, and it's even longer, 3.5 meters. So like 10.5 feet or so-ish. So 3 sections to the small intestines. Know the order, okay? And within the small intestine and large intestine, we have what are called Peyer's patches, and this is lymph nodes. So on Wednesday, I told you about the lymph nodes in the digestive system and how we have a lot of immunological surveillance. And so these lymph nodes are present within the small intestine and also the large intestine. There will not be any identification on this next quiz, okay? Unless you want to. I don't see a need for it. Okay, so within the small intestine, if we increase the surface area, we increase the amount of nutrients and water we can absorb into the bloodstream. Okay, it's kind of like the same idea with the respiratory system where the alveoli increased our surface area so we could maximize the oxygen. This is what's happening in the digestive system. We increase our surface area to maximize how much we can absorb, get as much nutrients as possible. Okay, so there are 2 major structures that are responsible for increasing your surface area. First we have is the villi. So if you cut open the intestines, it would kind of look like a towel. All those little loops, things sticking up like little fingers. So this picture here represents the villi. And so within each villi you have an artery and a vein and a lymphatic vessel as well. And so it increases the surface area. You have all these folds. If you stretched out those folds, it would be really long. So it increases the surface area. But there's more. So the villi have their own little fingers on them. They're microvilli. And so if this is the villi, we have these little tiny, tiny microvilli on a villi. So here is one, so villus is singular, and then the microvilli are the little tiny ones, each individual one on the surface of the villi. So you've increased your surface area twice. So huge amount of surface area thanks to your villi and your microvilli. Gotta know those, okay? Oh, and looks at this. Increasing surface area is 600-fold. A lot. Okay. Tissues within the duodenum. So, again, we know our alimentary canal is also lined with mucous membranes. We've talked about mucus and how important it is. So different cells that we're going to find within the duodenum. You have cells that are absorbing, so they're going to have microvilli. So villi, and then the little fingers off of that are our microvilli. So increasing our surface area allows us to absorb our digested food very well. Also, you do get some digestive enzymes. Goblet cells, they make the mucus. There are endocrine cells that make hormones like gastrin, secretin, well, not gastrin, that's in the stomach. Secretin, cholecystokinin, those are two examples that are produced in the duodenum. We're not going to worry about granular cells. Where are those? We'll just skip those, make our lives easier. Okay, slide 30. So looking at the last two sections of the small intestine, which are the longest portions, the jejunum is the middle, and then the ileum is the last portion of the small intestine. And what's really interesting is as you go, so the duodenum is the largest in diameter, and as you travel down the small intestine, the diameter gets smaller and smaller and smaller. Okay? And this is because as you travel along, things are being absorbed, and so you're having less and less material as you're getting closer to the end of the small intestine. So yeah, the diameter is smaller, you're going to have less villi, because most of the stuff is going to be absorbed before it gets to the end of the ileum. Okay, so remember, major site of nutrient absorption and water. Okay, Peyer's patches, mentioned those before. These are the lymph nodes you find within the inner mucosal layer and submucosa as well. Remember, immunological surveillance. Now, the section or the junction between the ileum and the ascending colon is called the ileocecal junction. There is a sphincter there that controls, it's a one-way valve, controls the movement of chyme from the small intestine, from the last bit of the ileum, into the large intestine, okay? And so it is controlled by the ileocecal valve, which remember is one way, and really important, why will it not write? Okay, there it goes, one way. And one reason for this is you have a lot of bacteria in your large intestine, which are beneficial, but the substances that are in your small intestine there's no bacteria, it's sterile. You do not want backflow, okay? You do not want bacteria from your large intestines backflowing up into your small intestines. That's bad. It does happen on occasion, especially when people are not getting proper medical care. Usually it's the elderly in rest homes. It can back up all the way. Not a good situation. But that usually is meaning they're not getting proper care. So this one-way valve is preventing bacteria from backing up into an area where it's clean, there's sterile, there's not bacteria. You don't want bacteria in your small intestine. You'll get sick. What is going on with this? Oh, okay, I don't know. Okay, moving on. So secretions in the small intestine. So, 3 important digestive enzymes. Disaccharidases, 1D. So, they're breaking down disaccharides, which are sugars, into smaller ones, so into smaller molecules, so into monosaccharides. Peptidases is breaking down peptide bonds, so that's between amino acids. Okay, so you're breaking down the bonds between amino acids, so then you can have individual amino acids, which are then easy to absorb through into the bloodstream. And then nucleases, well, kind of what it sounds like, breaks down nucleic acids, which is from your DNA. So anything you're eating is going to have DNA, whether it's plant or animal material, and so you need to break that stuff down too, and you can reuse those small components in your own body. Okay, and then the duodenum, we already know that secretin and cholecystokinin are secreted in the duodenum. It's stimulated by the vagus nerve. This is just a review. Chemical, so that's the pH, remember that? Or tactile irritation. So, for example, if the mucus layer was thin and it actually irritated it, then those glands would stimulate the secretin and the cholecystokinin. Okay, movements in the small intestine, 2 main movements. You have peristalsis, moving it in one direction, so propulsion, and then you're also going to have the mixing movements. So food just does not go in one direction in the small intestine. You do have mixing that's going on there because you have new digestive enzymes that are added in the small intestine. Okay, so segmental contractions are mixing contractions. Peristalsis is propelling. Okay, at the end of your small intestine, in the ileum, remember it separates the ileum from the large intestine. What separates is the ileocecal valve. Okay, that valve separates it. It stays contracted. See that? It remains contracted until peristalsis reaches it, and then it causes it to open, and whatever it's propelling, then goes into the large intestine. Okay, so when the peristalsis reaches it, allows the chyme to move into the first part. I'll show you a picture in a second, because you have your ileocecal valve coming in. You have this little pouch, and then your ascending colon going up. The little pouch, that's the cecum that it's referring to. Your little ileocecal valve would be right here. But there's a much better picture coming up. Okay, when you have chyme dumping into that little pouch, the cecum, it stretches, just kind of like your stomach stretches. It distends when you put adding stuff to it, and it will cause the local reflex. That reflex tells that ileocecal valve, okay, we got enough. Close up. There's only so much we can handle at once. So then the reflex closes the valve, no more chyme enters into the large intestine until that cecum is emptied. Okay. So, and then also, what I was talking about before, preventing backflow. Super important. So, let's see, where is the anatomy of the large intestine? Okay, well, we're just going to go back to the liver. Got to do the liver. Okay, so, real quick, with the liver, there are multiple lobes, and this next week you get to see the liver of your mink if you didn't already, if you were in, what, was it Tuesday lab? I don't remember which lab had the bad minks. Okay, what's really important about the liver, we're going to talk about the port, the hepatic portal, like the function of the liver itself. Yes, you have multiple lobes. I'm not going to ask you about those. But you have the hepatic portal, which is on the inferior surface. So this is the, there it is, shows you, inferior view. So you're looking at the underside of the liver here. So all of the blood that is absorbed nutrients from your small intestine, all of that blood goes to the liver before it goes anywhere else. And so that blood enters through what we call the hepatic portal. Okay? And so specifically hepatic portal vein. That is what's carrying the blood from the small intestine into the liver. Yes, you are also getting arterial blood, of course. There's nerves involved as well. You also have lymphatic vessels. So here, this is all kind of hard for you to see, but right here in the center, this, you see the blue one right here? That's your hepatic portal vein. That's what's coming from all of the blood from your small intestine, okay? Bile is produced by the liver and it's stored in the gallbladder, okay? And we're going to talk about these bile ducts in like the next slide. So I'm going to skip this part for now because we are going to talk about it in the next one, okay? So the bile ducts you will need to know, but I have a better picture for it to show you. Okay, this is the better picture. So here we have a view of the liver, gallbladder, the first part of the small intestine, so the duodenum, then the pancreas, and the spleen. Okay, so the stomach is removed, so this stuff is behind the stomach. Okay, so the hepatic ducts. ducts, so hepatic refers to the liver. These ducts, the hepatic ducts, are carrying bile, okay, to the common bile duct. So right here, you have a left and right hepatic duct, so one here, one here. They join together right here to form the common hepatic duct. Then it joins up with the much larger cystic duct. You see that? And then the cystic duct and the common bile duct join together. Yep. So the cystic duct and the common hepatic duct, join together to form your common bile duct. Yeah, you do need to know that, okay? So the common bile duct is what's carrying the bile to where it's going to enter the duodenum, okay? So what is interesting here is the common bile duct joins up with the pancreatic duct that's carrying digestive enzymes from the pancreas and dumping it into the small intestine. And so right here we have this joint area, it's called the hepatopancreatic ampulla, where the bile and the pancreatic juices are joining together and then empty out into the small intestine right here, okay? So there's a little sphincter that controls the bile and enzymes entering into the duodenum, okay? And both of those are very important in the digestive process. So remember, hepatopancreatic ampulla is where the bile duct joins with the pancreatic duct. And you also have this sphincter, I don't think that's mentioned here, that controls the movement of those secretions into the duodenum. Okay. And so the majority of the secretions are through what we would call the major duodenal papilla. Okay? Most of them are coming through that. That's the one I'm most interested in you knowing about. Getting a little more in-depth with the liver, like histology, cellular level. There's a lot of connective tissue in the liver. Similar to, there's a lot of connective tissue in lymph nodes and in the spleen that are kind of, wrapped around and then branch within the organ itself. And so it creates these little, they call them porta, or lobes, and then even smaller into the septa. There we go. And so a liver lobe is kind of like this honeycomb shape. So that's one liver lobule. Okay? So each lobule is going to have a center vein. Okay? So let me back up. So each lobule has a portal triad. Okay? And so the portal triad is kind of on the edges here. See these? So the portal triad includes your hepatic portal vein, hepatic artery, and hepatic duct. Hepatic duct is colored in green. Sorry, you can't see that very well. The hepatic duct is colored in green to represent bile. Okay, artery is carrying arterial blood. Hepatic portal vein is carrying blood that came from your small intestines. Then you have the central vein, this guy right here. Okay, that's in the center of the lobule. That's taking the blood that has been filtered through the lobule towards the inferior vena cava. So after the blood's been filtered through the lobule, toxins have been removed, other things have also occurred, then the blood can pass on, go to the inferior vena cava, and then enter the rest of the circulatory system. Okay, let's see. We're going to make life a little easier here. Okay, functions of the liver, most definitely you need to know these. So one very important function is producing bile. We need bile to help break down fatty foods, lipids in our foods. It helps break them down into smaller little pieces, and we'll talk more about what happens to the fat and the cholesterol once it's in the bloodstream. Also breaks down some other things as well. So what does bile do? Helps neutralize the acid from the stomach. Remember, so pH is very low in the stomach. So neutralizes and helps dilute it, helps emulsify fat, so breaking the fat down into smaller little bits. Secretin is secreted by the duodenum. Secretin stimulates bile release. Okay, and that will also so the bile secretions is going to increase the water. Bicarbonate naturally occurs, and that acts as a buffer. No, bicarbonate actually helps. Yeah, it's a neutralizing molecule, so it helps reduce the acid. Okay, liver is also very important in storage. So very important things that are stored in glycogen. Remember, glycogen is the easier energy molecule to break down. So short-term energy storage, you break it down, you have glucose released, awesomeness. Also store fat, which is also an energy storage method, but it's not as accessible. Vitamins, copper, and iron. Okay? Some nutrients are converted while they're in the liver. And one of the ones that's really kind of important is vitamin D. You have a small change to vitamin D called hydroxylation, where it turns the vitamin D into its active form. Okay? So that active form is really important. If we remember, vitamin D helps with the absorption of calcium in the intestines and has a lot to do with our blood calcium levels and bone health. Vitamin D and calcium close together. Okay? Detoxification is a really important, very important function in the liver. Hepatocytes, so hepato refers to liver, cyte means cell. And so hepatocytes specifically are kind of cleaning the blood, getting rid of urea, which also happens in the kidneys specifically. Removes ammonia, which is really toxic, converts it to urea, which is then filtered out in the kidney. Phagocytosis, so you have special cells that will get rid of dead, dying red or white blood cells and bacteria if it sees them. Those special phagocytotic cells are the Kupffer cells. Okay, so those are special within the liver. And then the liver does make things. And so we've talked about albumin, globulins, fibrinogen, were the three major proteins when we talked about blood that the liver produces. So heparin is another one that the liver produces, and other clotting factors. Okay, and all of these, so heparin has to do with, that helps prevent blood clotting. So people consider it a blood thinner, and clotting factors are essential in your body's ability to actually have your blood clot. So really important functions here. I never asked you guys to know this slide. Gallbladder. Did I miss something? Okay. You guys are signing the sign-in sheet? It's going awesome. Thank you. Okay. So I'm not too worried about the actual structure of the gallbladder. It is a sac that stores bile. The bile is made in the liver. Okay? So the bile is constantly being made, and it's dumped into that gallbladder, and it's being stored. And then it's released when you eat something that's fatty, or it's acidic, and you need to neutralize that acid in your duodenum. Okay? So it's stored, and it does get concentrated. So one of the things that stimulates the release of bile is cholecystokinin. The other one is secretin. I don't know why we don't mention all of these. So cholecystokinin, and then vagal stimulation, so the vagus nerve, so from the autonomic nervous system. Remembering that bile, you have right and left bile ducts come together, forming our cystic duct, joining to the common bile duct. Yeah. Gallstones. You've heard of gallstones. I just found out my mother-in-law, I found out several months ago, she had a gallstone. You know, usually they're not that big, but hers was like this big. Yes! I was like, how did you not notice there was something majorly wrong?
Did they have to make an incision to get that out?
She had surgery. Like, it could not be laparoscopic. They had to... It was a very big, oh my goodness, stone. So usually the major cause of gallbladder is precipitated cholesterol. So too much cholesterol that's building up in the gallbladder. It can block the duct from where bile is supposed to exit and then go into your small intestine. It blocks the duct. You have a buildup. The gallbladder gets inflamed. Not good. Gallstones can also be formed when someone undergoes drastic dieting. So if someone just goes on some crazy diet and they lose a lot of weight really fast, that can cause that precipitated cholesterol and builds up gallstones in the gallbladder and then can cause problems. So don't do a drastic diet. And I think you'll be okay. All right. This is a regulatory slide. Definitely need to know our regulatory information. So controlling the secretion of bile. Right. So we see our lovely diagram here. We've got a lot more going on. We have cholecystokinin, secretin, and bile. So let's see what happens. So we're going to start at number one. Number one right here. So the duodenum produces secretin when the pH is too low. So the chyme coming from the stomach is too low. Okay? That secretin is going to stimulate. So number one here. Oh, I'll do it in purple to match. So number one, our secretin is going to go and stimulate the liver to secrete bile, okay? In addition, we have cholecystokinin. I'll try to do it, let's see, hot pink. There we go. We have cholecystokinin, that's number two right here. So cholecystokinin is also produced by your duodenum, and cholecystokinin works directly on the gallbladder. So it travels through the bloodstream, and then its target tissue is the gallbladder, stimulates the gallbladder to contract, so squeezing the bile out, which then passes through those bile ducts, okay? And so that will release the bile into the duodenum, okay? So as a reaction to cholecystokinin, the bile is released, okay? So secretin tells the liver to secrete bile into the gallbladder. Cholecystokinin tells the gallbladder to squeeze, contract, squeeze the bile, which then enters your duodenum right here. Okay? Next, number three, vagus stimulation. That's orange. That's up here. The next thing that happens, vagus nerve stimulation also tells the gallbladder to contract. Okay? So we have cholecystokinin telling it to contract. We also have the vagus nerve telling it to contract. So two methods to get bile into the duodenum. Let's see here. Bile salts. So that is in the blue. It's like a light blue. So bile salts right here, number 4. Okay, so bile salts that are in your duodenum can also trigger more bile secretion, okay? And so, same deal. So you have 3 different mechanisms that trigger the secretion of bile into the duodenum, and they're all working together, okay? So it's very important. So know the control. So what does cholecystokinin do in the role of bile secretion? What does the vagus nerve do? What does secretin do? Okay. Moving to the pancreas. Pancreas, I remember when we talked about the endocrine system, the pancreas is a dual function organ. It is both an endocrine organ and an exocrine organ. So endocrine, it makes insulin and glucagon, and I think it also makes one other thing. Can't remember right now. Oh, somatostatin. Okay, those are hormones. But then the exocrine portion of it is making digestive enzymes. Okay, and those are what are passing through the pancreatic duct into the duodenum with the bile. Okay, so first just looking at the anatomy, the pancreas has a head, body, and a tail. Okay, so the tail is over here. Your head is this portion, and then the body is the rest. Okay, so exocrine and endocrine. So let's see. Endocrine first is made by cells within what we call the pancreatic islets, little tiny islands. So the three hormones, you should know the hormones that are made. I'm not going to ask you which cell produces which hormone. You did that in the endocrine system. The exocrine portion of the pancreas, they are in what we would call, so first of all, pancreatic islets, these are the blue stuff, the blue little round ones here. These little lobules outside of those are the Ancini cells. Those are what are producing your digestive enzymes. So if you look closely at the picture, can we look closer? So this pancreatic islet has a connection to the circulatory system. So that would make sense. It's endocrine, so it's going to travel directly into the bloodstream. The Ancini cells that are making digestive enzymes travel in ducts and then join together to the pancreatic duct and then empty it into the duodenum. Okay, so they're traveling in different ways and they're affecting different things. Don't worry about this part right here. We don't want to. You should know, yes. So looking at this bottom picture a little closer, if I can get it. So I'm going to erase my blue. Pancreatic duct. So right here, I'm going to do it in purple. This is your pancreatic duct. You see how you have the smaller ducts that come in and join together. And so it's collecting all of those digestive enzymes. They all come together at the pancreatic duct, and they join with the common bile duct, and right there at the hepatopancreatic ampulla. Okay, and remember, please, that the bile and the pancreatic digestive enzymes cannot enter the duodenum unless the hepatopancreatic ampulla sphincter allows it to. So there is a sphincter that controls the release of the bile and the enzymes. And so when the sphincter releases, then it is released in through the major duodenal papilla, this little hole, into the duodenum right here. Okay? So please make sure you know this. Okay, you need to know what the hepatopancreatic ampulla is. You need to know that it's this is where your pancreatic duct and your common bile duct come together. And right before that sphincter, which controls the flow of the bile and the enzymes into the duodenum. Okay? Okay, so talking about pancreatic secretions, let's see here. Some things we do spend more time on, some things we do not. Okay, this is an important one here. Enterokinase, what it does is important. So activates trypsinogen, converts it to trypsin, and we will come back to that later. Okay, so you do need to know that. I'm not going to... Oh, and then trypsin activates, so there's a whole chain reaction. Tryptin then converts this chymotrypsinogen to chymotrypsin. And also converts the procarboxypeptidase to carboxypeptidase. All three of those, what's the point of that? All three of those break down proteins. So they are proteolytic. So really, right here, this tells you the three main dudes, the activated things, trypsin, chymotrypsin, and carboxypeptidase. All three break down proteins. The process of breaking down proteins, or the ability, is called proteolytic. Okay? Proteolytic enzymes. I still have 7 minutes. I know one of you guys are packing up. Also releases amylase. So remember, amylase was in our saliva, but then it was inactivated when it got to your stomach. But then once the chyme is in your intestines, amylase is released again. Okay? Because amylase does not work with acid. So, but we don't want it really acidic in our small intestine, so then the pancreas can release amylase again and continue that carbohydrate digestion. So starches, carbohydrates. Lipase is also made by the pancreas, and as it kind of hints, breaks down lipids as well, helps with the bile. And then we have deoxyribonucleases and ribonucleases, and both of those have to do with DNA or RNA breaking down. Okay, controlling the secretion in the pancreas. I know this is so fun for you. So, hint for studying. These slides where they're talking about the control of secretions, these are good ones to study, good ones to quiz each other on, like, okay, this happens, what's next? Okay, so secretins release, what happens when secretins released? Or gastrin's release, what happens when gastrin's released? Okay, so we want to know the potential effects of all of those hormones. So this is why I think maybe I'm making a table of each hormone and its functions, what stimulates it, would be really helpful. Okay, and what action it has. So secretin, when we're talking about the pancreas, so it tells the pancreas to secrete bicarbonate. Bicarbonate helps neutralize acids, okay? Which we don't want acid in our duodenum, so it helps with that reduction of acid. Cholecystokinin, its effect on the pancreas, so it tells the pancreas to release lots of digestive enzymes, okay? The vagus nerve, coming from the brain, tells the pancreas to also release digestive enzymes. So, noticing here that both secretin and cholecystokinin are released into the bloodstream, travel back around and tell the pancreas to release those enzymes. It's kind of circuitous. That's the word I wanted to say, but it works. Okay, large intestine sections, and this is what I wanted to show you before, but it was a little far down on our slides. So, here is the last part of the small intestine, the last little tiny bit of the ileum. The section between the ileum and the first part of the large intestine is the ileocecal junction. And so here, that's where that term is right there. The ileocecal junction is right here. And in that junction, you have the ileocecal valve, which remember is a one-way valve controlling the movement of chyme from the ileum into the large intestine. So notice, if you have material that's coming in right here, there is that little pouch. There's that cecum. Okay? It's a little pouch. That distension, remember, then causes a local reflex and tells that valve to close so we don't get backflow. Also notice that at the bottom of the cecum is our appendix. Has a little bit of an immune function, not much. So if it does have to be removed, you're going to be just fine. Okay, so cecum is this little portion here. Then traveling up is our ascending colon. Ascending is retroperitoneal. Transverse, not retroperitoneal. Descending, retroperitoneal. Then you have the sigmoid colon, and here, sigmoid colon, kind of think about it as an S, okay? So you got this S shape, sigmoid is an S, and then your waste collects in the rectum, and then you have two sphincters that control the when things will exit the rectum through the anus. So you have an internal sphincter and an external anal sphincter. Okay. So notice movement within the large intestine is slower, and this is where there's a lot of bacteria. Helps with the breakdown of products we can't break down. Movement through there is slow. There's lots of mucus, otherwise you would have some painful problems. You'd get some compacted intestines there and some really awful constipation, painful defecation. You don't want any of that. And so you do have water being reabsorbed. Bacteria also produces vitamin K. Okay, so water's getting reabsorbed, vitamin K is being reabsorbed. What else? What did I miss? Oh, salts, so like ions, like sodium, calcium, stuff like that. So by the time you're at the rectum, 90% of the volume that entered into your large intestine has been reabsorbed. So don't get this confused, though. The majority of nutrients and water are still reabsorbed in the small intestine. Whatever is left is then mostly reabsorbed in the large intestine. So you are conserving water as much as possible. Okay, that is a goal in the intestines. Oh, we didn't finish. Okay, so we'll continue on with the anatomy. We're almost done, so we'll finish this Monday and start on urinary system Monday. So start planning on that. Get some good studying done and some good rest this weekend. Happy Easter in advance. I hope you're able to go see the Easter pageant. What is it called? It's not a pageant. It's the passion. Yeah, through the church. They did it last week and they're doing it this week. Yeah, so the whole, the last days of Christ and rising. And I hear it's very good.